All right, so we've talked about the retina at the back of the eye. It's just a layer of neurons, a layer of cells. The most important are those photoreceptors, the rods and cones, but we mentioned those go through a pathway through some other cells before they leave the eye. So the rods and the cones go to some bipolar cells, kind of just passing the signal along. Then they go to ganglion cells, specialized type of neuron. They're really large cells. Those then send the signal out the back of the eye, out the optic nerve. So we're going to kind of follow that now and see where that leads in the brain, and then we'll kind of see how these cells work along the way for the whole visual pathway, eventually building up to specialized cells within the brain that might help us process, faces, recognizing a person we've seen before, a house, a car, a tool, a chair, whatever it is. So the signal is going out the eye here. So you can see kind of the optic nerve leaving the eye here. And it actually, half of each of those optic nerves crosses over to the opposite side, while half stays what we call ipsilateral on the same side. The other half goes contralateral to the other side. So we'll come back, we'll talk about that in a moment. But where does that information go then to get to actual processing, to actually start having a visual experience? It's going to pass through one area before it gets to any cortex, before it actually gets to the cerebral cortex, the main lobes of the brain. So first it's going to pass through the thalamus. And you may remember the thalamus, there's actually a copy on each side, but the thalamus, that's our sensory gateway. It's where information has gone through for all of the senses we've seen so far. In fact, all of our senses except for smell pass their info through the thalamus. In this case, it's going through a layer called the lateral geniculate nucleus. You can just remember it as LGN. We met a very similar structure when we learned about audition and how the signals, as they go from our ears towards A1, the auditory part of the brain, they pass through the thalamus, and we saw that they pass through the MGN, the medial geniculate nucleus, which just means towards the middle of the brain for that chunk of the thalamus. This is the lateral part, so towards the side of the brain, lateral geniculate nucleus. But the point is they pass through the sensory gateway, basically some layers that help pass along the visual information, and we'll see kind of the purpose of that soon. But then where they go from there is to end up in what we call V1, the primary visual cortex, which is just at the very back of the occipital lobe. So you may remember from when we learned about the lobes, that sort of primer on neuroscience stuff, the occipital lobe at the back of the brain is a huge area primarily devoted just to vision. And it's at the very back of that we find V1, what we'll call V1, just like we had A1 for the primary auditory cortex. This is our primary visual cortex. We had S1 for primary somatosensory cortex. You kind of get the picture now. Now, the visual information, as I said, some of that crosses over to the opposite side of the brain. And the reason it's wired this way is not so that each eye's information gets processed on the opposite side of the brain, because that's not true, right? Some of the information that hits our left eye still goes to the left hemisphere of the brain, and some of the info hits the right eye still goes to the right hemisphere of the brain. When we say that vision is processed contralaterally, remember that's our opposite side processing, what we mean is the left visual field, the stuff in front of this guy's face, the visual field, what he's seeing off in his left periphery, that information will all end up in his right hemisphere of his brain, and vice versa. Things that are off to your left in the visual field, like we can see here, kind of in these blue lines, things that are off to your left, they're going to come in at such an angle that for each eye, right, they're going to hit both eyes, but for each eye, they're hitting sort of the right wall of the eye, the right wall of the retina there, which those wires, as you can see, the right wall of both retinas has the wires coming out and crossing to or going to the right hemisphere of the brain, which means any info that came from the left visual field, anything you're seeing off to your left, will end up processed on the right half of the brain. Anything you're seeing off to your right, if there's a picture of a car over there, if you're seeing something flash by real quick, a tiger's approaching you, that information, just based on the angle where it hits the retina and where those wires go, you can see it will always end up on the left hemisphere of the brain. So when we say contralateral processing for vision, it's not that each eye is processed in the opposite hemisphere, because both of our eyes will get information from the same part of visual space, right? They'll often overlap, but it's the angle it comes from, which means the part of the visual field gets processed contralaterally. Here's just another picture of that so we can see the overview, make it a little more clear. So things that are off in your left periphery, right, they're going to have both of your eyes, but they're going to go, the part in the green here are going to go to the right half of the brain. The part in the yellow here is going to go to the left half of the brain. And once again, you can see after this little crossover here, the optic chiasm is where they cross. The important thing is it goes through the thalamus, the LGN of the thalamus, before getting back to V1, the primary visual cortex. All right, so that's the pathway. What I want to talk about in this video, the main thing, is that idea of receptive fields that we've talked about before, which you'll remember the definition is just for any given neuron, it's whatever pattern of stimulation affects that neuron's firing. So whatever makes that neuron go off or makes that neuron not fire as much, right? You can have an inhibitory effect on a neuron, but it's the kinds of input, the kinds of things that can happen, the stimulation that can cause that neuron to do something. So for example, if there's a neuron in your brain that only fires when you see a square shape off in your far right periphery, then we would say that neuron's receptive field is square-shaped light patterns hitting that part of the retina at exactly that angle, just because that's the type of thing that makes this neuron fire. It only fires for that thing. So if I find a neuron deep in my brain that only fires when I'm looking at Jennifer Aniston, I'll call it my Jennifer Aniston neuron, then that would be the receptive field of that neuron is visuals of Jennifer Aniston. And yes, there's a published paper about Jennifer Aniston neurons and grandma neurons and all this stuff we'll talk about later. But we're going to start very, very simple. We're going to start with the neurons early in visual processing, which actually have very simple receptive fields. The things they fire for are very simple inputs. So starting at the very start, the receptive field of a rod, right, just a simple photoreceptor. Remember, we have rods and cones. Those go off for certain wavelengths of light. So the receptive field of a rod is just light hitting that little bit of retina that it's on, light that hits that part of the retina where the rod exists. Specifically, it has to be light of the appropriate wavelength because rods only go off for certain wavelengths of light, as we see. But as long as it's the right wavelength of light hitting that little bit of retina, that's what makes this rod fire. So that's its receptive field. So kind of the rod just gets turned on or off if light hits it. Then if we look at a cell a little later in that visual processing pathway, so we still haven't left the eye, we're still back there in that retina at the back layer of the eye, but now those ganglion cells that are hooked up to a bunch of rods, we could say, what's the receptive field of a ganglion cell? What determines whether a ganglion cell fires? And we already saw a little of this when we did that kind of practice looking at convergence of a bunch of rods might be hooked up to a single ganglion cell. And we might model that with just a few, like 5 or 6 rods hooked up to a ganglion cell. But in reality, a ganglion cell could have hundreds of rods hooked up to it. In fact, it's pretty common for 100 rods to be hooked up to one ganglion cell, as we can kind of see in this diagram on the right here. So when we're asking what makes this ganglion cell fire, what makes it send its signal off to the brain that says, oh, I got some light hitting the area I'm watching, well, we look at what it's wired to. It's wired to 100, we'll just say around 100 rods here. That's what each of these little bits, the green and red bits here, all in this kind of big circle pattern, those are just a bunch of rods all hooked up to the ganglion cell. And if enough of these fire, or if they fire in the right combination, if the right ones fire, then they're going to send a signal to that ganglion cell. Enough of a signal will get through that'll make the ganglion cell fire. So basically, the ganglion cell needs enough total input from all those rods it's hooked up to fire to the brain. So what really is happening is that ganglion cell is watching a larger chunk of the retina. A larger chunk meaning that combined area where all those rods are, that's the area where if light lands, it might affect the firing of this ganglion cell. Now, that's just if all of these rods kind of gave a simple excitatory input to the ganglion cell, and we were just looking for enough combined input, enough combined excitatory input to make that ganglion cell fire. But it turns out ganglion cells are wired with a little extra complication. We say that they have a center surround receptive field, meaning that these rods in the middle here, the center of the receptive field, those rods that we've highlighted in red have an excitatory connection to the ganglion cell, meaning if light falls on these rods and makes them fire, it's going to make the ganglion cell itself more likely to fire. But if light lands in the surround, right? The sort of ring of rods that aren't at the center, these ones in green here, those have an inhibitory connection to this ganglion cell. I mean, if light falls there, it actually makes the ganglion cell less likely to fire, or it'll fire at a lower rate. So that's why we say ganglion cells have a center surround receptive field. So you can think of it kind of modeled out here, right? If the center is our excitatory input and the surround is our inhibitory input, then imagine some light falling on the receptive field. Remember that just that area of the retina that has a whole bunch of rods all next to each other. It's literally just a circle of area on your retina. If some light falls, let's say just a tiny pinpoint of light falls right in the center of that excitatory center, it's going to hit, the 10 rods in the very middle there. It'll cause a little extra excitement. So down here, this would be our spikes, our neural firing, the action potentials. And this is a little higher rate than whatever the baseline of this cell was when it's resting. So it causes a little bit of excitement. Now, if a little bit of a larger swath of light falls there and it falls entirely on that excitatory center, then we're going to get this cell going off, the ganglion cell. It's going to be firing like crazy. So the signal going to the brain will be pew, 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 like firing very fast. However, if that light is a little wider, now it's hitting all that excitatory, but also quite a bit of inhibitory area, so that'll make it not fire as fast, right? It's got some minuses to cancel out a lot of those pluses. And likewise, if it's a totally diffuse light, right, that would probably be hitting a bunch of other areas of the retina nearby. In other words, light that is not specific to this ganglion cell, it's just a big wide light everywhere. Well, then it's going to have a lot of inhibitory input to cancel out most of that excitatory input, and it's basically going to fire, similar to its baseline, maybe even a little lower than its baseline. So that's how a ganglion cell works. Its receptive field is center surround. What it's trying to do is get very specific about where the light is falling, even though it's hooked up to a bunch of different rods. That convergence gives it a lot of sensitivity, as we saw, so that's very useful for picking up even faint bits of light by combining across an area, but we still want to be pretty specific about what that area is. So the center is its way of being specific, and that inhibitory surround is its way of making sure we're not going to mix up a situation of a specific point of light where I know it's hitting around here versus a wide diffuse light that we don't want that ganglion cell to be pretending it's the only thing getting light, or it knows exactly where the light is. All right, then that signal goes out from the ganglion cell. Remember, it goes out the optic nerve, it goes into the brain, kind of deeper into the brain, and before it gets to the cerebral cortex, that important outer layer of processing where we actually start to see something, the information gets passed through the thalamus, the LGN, lateral geniculate nucleus of the thalamus. What is the receptive field of a neuron there? You can kind of think of it like an individual ganglion cell just hooks up to an individual neuron in the LGN, in the thalamus, and it just passes that signal along back to V1. So in that case, the LGN neuron, what makes it fire is just whether the ganglion cell fired. And since the ganglion cell had a center surround receptive field, it was watching that little circle of the retina and going off in that particular pattern, that center surround pattern, then the neuron in the LGN will have the exact same pattern, because it goes off if the ganglion cell goes off. It doesn't go off if the ganglion cell didn't go off. If the ganglion cell goes off a whole bunch in a row, the LGN cell that it's connected to will go off a whole bunch in a row. So you can kind of think of those as having the same setup, because the LGN is kind of just passing that along. Now, why then do we go through the LGN at all? Like, why doesn't the wire from the optic nerve just go all the way directly back to V1? So let's just take a quick aside here and explain why that exists, and really why the thalamus exists in general, why all of our senses, except for smell, pass through the thalamus. Well, the LGN, and so remember, this is just a chunk of the thalamus here, it actually gets a ton of input, not just from the eye, right? Not just the incoming information from our eye that needs to get passed on to the cortex part of the brain, where V1 would be, but also the size of this large arrow here is to show us there's a lot of neural connections coming in toward the LGN from other parts of our brain, from the cerebral cortex itself. So we actually get a lot of input and feedback from other parts of the brain to help kind of tune the LGN before it even gets the info from our eyes, which helps filter out certain things, limit what information goes through, or adjust or emphasize or de-emphasize which signals are getting passed on to V1. You can think of this like For example, directing your visual attention towards something. Like I might have my eyes looking straight forward, but I'm paying attention. I'm at the club or something. I'm watching this hottie off in my left periphery. So my attention is covertly directed off to the left part of my visual field, even though I'm not staring at the person, right? It's just in my periphery. But my attention is there. That is something my brain has chosen to direct my attention over to that part of the visual field, which means things off in my periphery that normally might not get processed very heavily because they're kind of just the background stuff off to the side, that normally I might not even notice or have a conscious perception of. Well, if I'm attending to that, then what's going to happen is my brain says, hey, pay some extra attention to those signals. When that comes in from the eye, from that part of the visual field, actually send all that stuff through to the cortex. I really want to pay attention to that and process that heavily and maybe even have an experience, an actual conscious perception of what's going on over there, where otherwise I usually wouldn't notice things in my periphery, and that info just disappears after a moment. So that's kind of the idea of the LGN. It allows for a lot of top-down influences or information coming in from the cortex that affects perception. Because, again, V1, that chunk of our brain back there, it isn't going to be able to pay full attention to deep processing of every little bit of information that comes in from every angle of our visual field. We get way too much information just in any given moment of our lives. Imagine you're looking out at a crowd of 10,000 people in a stadium. Everyone's wearing a different color of shirt, has different hair, whatever. Just because that information hits your eye doesn't mean that it needs to be processed in your cortex. We actually ignore most of the information coming in, and it never gets processed very deeply. It doesn't really get to the part of our brain, the visual processing of our brain that has us see in the sense of having a perceptual experience. So we don't see most of what hits our eyes in that sense, because things like the thalamus here will choose what we're paying attention to at any given time based on input from the cortex. So that's the role of the thalamus. That's why it goes through this sort of extra layer of filtering to help us direct where our limited processing power goes. All right, from there, from the thalamus, as I said, it goes back to the cerebral cortex, and where it ends up is the real place where we could say visual processing really starts for real, and that is the primary visual cortex, so V1. Main visual processing center. Of course, it has to send this information on to other parts of the brain, even just other nearby parts of the occipital lobe, so that we can take these simple parts and kind of reconstruct from it the entire visual experience, putting together angles and shapes, and from that putting together information about color and about movement in the visual field and things like that, to have the full experience of what we're recognizing or how we're using visual information to accomplish things. But here, things still start pretty simple. So we're going to find neurons back here in V1 that have specialized receptive fields, specialized things that they fire for, but it's still pretty simple stuff. So for example, a lot of neurons back there in V1 only fire for lines, what we might say sort of edges of light, which is another way of saying a contrast, a line of contrast in the visual field where things go from a little lighter to a little darker, where they change color. Look at the edge of your room, and as you go from the floor to the ceiling at the edge of your room, there's a line there, right? That's the corner of your wall. Well, that is itself an edge that's going to be noticed visually. It's just slightly different colors of light, slightly different patterns of light hitting your eye in a straight line. That's the kind of thing that turns on cells back there in V1 in your primary visual cortex. There are some other neurons back there too, but this is actually a very common type. This is sort of the basic building block of a lot of our visual perception, is just little lines, little edges of light or of contrast and light. Now, how do we know how the V1 works, or that we have neurons back there that only fire for something like a little line, a little edge? Well, we actually know this from some work that was done back in the 60s and the 70s by David Hubel and Torsten Wiesel, who got a Nobel Prize for this work, by the way. It's very important stuff. And they did it in a way that, again, involves animal models, so maybe not something we'd all be comfortable doing. It's kind of messed up in the sense of what the animals have to go through. Of course, they don't, there's no pain involved or anything. They use anesthesia or analgesia to make sure that they don't feel pain during any surgery and things like that. But what they do, or what they did back in the 60s and 70s in this series of very famous studies, is they used cat models initially. And with these cats, they would set them up in front of a screen. So they're looking forward at a screen. And they need to make sure that the cats aren't moving their eyes around, looking at different parts of the screen, because then whatever line or whatever dot or whatever light that's being shined on the screen, whatever pattern is being put on the screen, if the cat was just looking at that thing, then it would always be coming in at exactly the angle that hits the fovea, that hits the part of the retina of whatever we're looking directly at. But they wanted to know also how cells process things off in our periphery, things up and to the right or down and to the left. So what they did is they actually temporarily paralyzed the cat's eyes to make it so that the muscles that normally move our eyes left and right don't actually work for the cat. It's a temporary thing, but they temporarily shut that down so that the cat is staring directly at this fixation point right at the middle of the screen and can't look to the left, can't look to the right. So we know for a fact exactly where it's looking, and thus if I do something up here at the top of the screen, I know it's going to come in at just the right angle to hit the bottom of the cat's retina that processes things in its top periphery. Okay, so now we can present things in a controlled manner to different parts of the visual field in front of the cat, knowing exactly where it'll hit the retina, exactly the angle it comes in, and see what happens in the cat's brain when different visual things are in front of it. So what they did, it's pretty simple stuff, is they just shine different patterns of light onto the screen in front of the cat in an otherwise dark room, while recording from an individual neuron back in V1, or the equivalent of V1, the homologue of V1 back in the cat's brain. So they're back there in V1, just clamped on with an electrode measuring a single neuron. They just pick a neuron sort of at random to see what makes this random neuron fire. What is it that this neuron is watching for that makes this neuron go off? And what they found, let's say they just plug into one neuron, when they tried different things like bars of light, for example, at some points this neuron would go off. It would go crazy. But the exact same stimulus, if you just tilted it a little bit and made the bar horizontal instead of vertical, even at the exact same place in the visual field, that neuron wouldn't fire. It wouldn't give a crap. It wouldn't do anything. As you get close to a vertical line in that specific part of visual space, this one neuron goes off. But if you take that vertical line and present it somewhere else in the visual field, the vertical line does nothing for this one particular neuron. So they found, here's this one neuron that fires specifically for a vertical line, so a line of a certain alignment, a line of a certain angle, at a particular part of the visual field. And nowhere else, for nothing else, does this neuron fire. It has a very specific thing that it goes off for. So we'd say that's its receptive field, right? That's all that receptive field means, is what does this neuron fire for? So back there in the back of your brain in V1 is a neuron that only cares about a little vertical line in this part of your visual field. And somewhere else, we might find a neuron that only fires for a line that's at a 45-degree angle in the upper right part of your visual field. And another neuron that only goes off for horizontal lines in this lower right part of your visual field. And another neuron that only goes off for vertical lines in this lower right part. And another neuron that only goes off for 45-degree angle lines in this part of the visual field, and so on. But covering the entire visual field for every possible combination of angles in each place. That is what those V1, they found sort of a V1 neuron for each of those. So these types of cells that just look for a line of a particular angle in a particular place, those they called simple cortical cells. And cortical here just means there are cells in the cortex now, right? We've moved from rods and ganglion cells, which were in the eye, right? The retina in the back of the eye. And we've moved through the thalamus. That is a subcortical structure. It's before you get to the cortex. It's under the cortex. Now in V1, we are actually in the cortex, in the lobes of the brain, the big outer layer, the cerebral cortex. That's why they say cortical. But sometimes this is just shortened to simple cells. So in V1, we have a bunch of simple cells, neurons that have what turns out to be basically a side-by-side receptive field that makes them fire only for lines of light in that particular orientation. So for that vertical line, it's set up so that it fires maximally if the light falls exactly in the place it's looking, right? Exactly in that excitatory bar there. But just like our ganglion cells had that inhibitory surround, these simple cells also have an inhibitory sort of side-by-side surround. So if the light falls a little too much to this side or a little too much to this side, then the neuron that's very specific to this one vertical little strip of the visual field wouldn't go off quite as much if you moved the light over a little bit. Or if the light is just diffuse, right? If it's a big wide light that covers all of this, it's going to land on the pluses and the minuses if it was covering this whole thing, but then the minuses would cancel a lot of that out and the cell wouldn't fire as much. So it gets maximum firing if there's specifically a sort of edge of light or an edge of contrast, a little line of contrast right there in the visual field, not to the side, not to this side, but right there at exactly that angle. You get maximum firing of that cell. A bunch of action potentials going pew, 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 pew. But if the light's tilted a little bit, so not just in the wrong part of the visual field, but even if it's just at the wrong angle, then of course you can see what happens is it's still hitting a lot of the excitatory part of the receptive field, but it's also hitting a lot of that inhibitory part too. So it cancels that out. You don't get as much firing. So the orientation is wrong. And of course, the more you tilt that, the more you're getting into the inhibitory and the less of the excitatory. So the maximum firing is always specific to that straight direction, whatever it is, whatever angle. And another one might be specifically looking just for that tilted angle, but that means its inhibitory part would be tilted to the side there too. So those are simple cells. Now, each of those simple cells in V1 is hooked up. It's getting input from a bunch of LGN neurons back there in the thalamus, the stuff that sent its input to V1. There's basically some convergence going on where a bunch of those LGN cells, so this is here in the thalamus, the orange would be the LGN cells, a bunch of those are wired up to a single simple cell in V1. Remember, this is our detector here in the back of the brain in V1 that detects lines. And those lines you can kind of think of as being made up of a bunch of these little center surround circles, the things that the ganglion cells detected, which of course passed their signal to the LGN cell, which then has the same center surround receptive field. So basically, if there's a bar of light at the exact correct angle, the angle that this simple cell back there in V1 is looking for, so we found this V1 cell that only goes off for 50-degree angle of light, right? A line that's at 50-degree angles in that particular part of visual field. What's happening is the way to get that to maximally fire is to actually have some light going at that around that 50-degree angle, because that's going to turn on a set of ganglion cells that are all in a row, right? Kind of partially overlapping with each other, but it's hitting all of their centers, which is the most you can do to make those go off. And then those are hooked up to these particular LGN cells, which are hooked up to that V1 cell, which means that V1 is just looking for a combination of that earlier stimulation, a combination of ganglion cells going off, which is another way of saying a combination of those hooked up LGN cells going off. That's what it is to detect a line. That's what it is for your brain to know that there's an edge somewhere in the visual field, is just a bunch of ganglion cells went off. The correct set of ganglion cells that it's hooked up to went off. But if the line of light, the edge we're looking at, is the wrong angle, right, then it's going to turn on maybe a couple of these ganglion cells quite a bit by hitting a lot of their center. But these other ones that this V1 cell is watching via the LGN cells, those aren't really going off very much, which means the LGN cells aren't really going off very much, right? The outer ones, just these middle two, which means this V1 cell won't go off as much. It's not going to say, hey, I detected a line at my angle that I look for. It's not going to fire a lot. Whereas up here, it's saying, yep, I found a line, I found a 50-degree angle line. So that's kind of how things are set up so that these simple cells can detect a line. It's not magic. It's just looking for the right combination of simpler receptive field input from previous cells. And that's how you build a cell that has a more complex receptive field, like a line. This is just another example of that same idea. So again, imagine a person looking forward at a screen in front of them. That's their visual field. We might have a neuron with that sort of straight line, a simple cell, so it's got a straight line, in this case for an angle roughly like this. That's its receptive field. So so let's say it's in the upper left of the visual field, then if some light falls there right in the right place, it is going to fire. But if that same angle falls somewhere else in the visual field, like off to the person's right, it doesn't work. Off to the person's upper right, it doesn't work. Something could be in the correct place in the visual field, but be the wrong angle, not going to fire. The correct angle bottom left doesn't fire. And maybe you got the correct angle with a line that does overlap somewhat with what we're looking at and also hit some other areas, then it might fire a bit. And basically the brain can look for patterns of different simple cells going off to know where we're seeing various lights in the visual field, various lines of light or lines of contrast, right, edges, which can allow us to look for things like a square pattern, right, which is just 4 lines all going off, or a house, which is just 4 lines going off and then kind of a triangle shape at the top, right? So combinations of simple little bits of line, bits of edges can be added up to make more complicated structures that a cell later could fire for if it sees the right combination of earlier cells going off. Now, if we look at V1, so back when Hubel and Wiesel were doing that thing, like poking in the cat's brain and picking out individual cells in V1 and seeing what they go off for, what we find is a very organized map of how these V1 simple cells are laid out. So there are sort of simple cells that go off for a particular angle, right, a particular orientation in the visual field, and they're all next to each other. So all the cells that go off for a 45-degree angle will all be literally next to each other back here in the brain. This is just just layers and layers and layers of neurons. So a complex 3-dimensional structure of neurons back here in V1, but all of the 45-degree angle ones are next to each other. So we call that an orientation column, right? It's a whole column of neurons that all go off for a particular orientation. Likewise, if we come in sideways and look at layers of these neurons here, one after another, what we find is for a particular part of the visual field, we're going to find the neuron that goes off for horizontal lines next to the neuron that goes off for slightly horizontal lines, which goes off for angled lines, next to the neuron that goes off for more angled lines, next to the neuron that goes off for vertical lines, next to the neuron goes off for the next angle, and so on. So basically, for a particular part of the visual field, we've got all the neurons that detect different angles of light in that little spot of your visual field. All of those neurons are actually next to each other in the brain. So what we have is this giant 3-dimensional map of all the possible angles and locations laid out in a very orderly way. So we call it a retinotopic map. It's organized based on the layout of the retina, or the layout of the visual field in front of us. It's not random. Rather, just like A1 had a tonotopic map and S1 had a somatotopic map, remember that body map, that homunculus, now we have a retinotopic map, a map of different parts of the retina, and in this case for specific angles of lines at each of those parts. It's laid out in a very structured manner. So what's the point of different neurons having different receptive fields? Like, why are we talking about receptive fields? A neuron that fires for different things. Well, that allows it to act as a filter. So different neurons fire to different input. That means the brain can then combine all these inputs, like we saw with that V1 cell, firing only for specific combinations of earlier, simpler input, that we can actually start to recognize more complicated things by looking at for combinations of simple things. So I can have a neuron that is tuned to only fire for a certain combo of earlier inputs. That means I can recognize more complicated parts of, objects and whole objects and whole scenes made up of objects, and have neurons that only go off for that specific thing. Neurons that only fire for when I'm looking at chairs or cats or whatever. So it starts with detecting very simple features, but the brain is going to be reconstructing that input from those earlier cells, which again are just a bunch of pew, 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 pews coming in from those individual neurons. It's all electrochemical signals, but it's going to reconstruct that input by looking at what combo has gone off to allow us to perceive more complex features and recognize actual things, that we're used to thinking of in reality around us, cars and stuff like that. All right, we'll stop there for now. We'll see some more complex cells and get into more complex parts of the brain, recognizing that higher level stuff in the next few videos.